In this video, I will talk about ion exchange chromatography. This is a special kind of chromatographic technique. Like other chromatographic technique, we have two phases, stationary and mobile. Stationary phase is the solid phase and mobile phase is not the solid phase. It can be either liquid or gaseous. Now, how ion exchange chromatography is different from other chromatographic technique? In this type of chromatography, size of the particle doesn't matter. Only the charge on the particles matters. So with this, ion exchange chromatography is of two types, cation exchange and anion exchange. So let's get started with a brief discussion about this technique. This technique relies on the attraction between oppositely charged particles. Purification of components is based on ions. Any charged molecule like protein and nucleic acid can be purified by ion exchange chromatography. Two phases, as I have mentioned, mobile phase and stationary phase. So in this chromatographic technique, charged molecules like proteins and nucleic acids can be purified. So let me show you with the diagram. This is the column and this column is filled with the stationary phase. This yellow color is the stationary phase, like resins. So this stationary phase is porous in nature, that is, it has beads through which the mobile phase will be eluted or pass. So this column contains stationary phase like resins. Here we have the reservoir, and this reservoir is filled with sample, that is protein or nucleic acid to be separated, plus buffer solution of desired pH. Now, if this reservoir containing containing the sample and the buffer is poured in this column, so this column, let me tell you, contains stationary phase like resin, and also it is filled with charged particles. Let's say this is filled with negatively charged. This resin is embedded with negatively charged particle. Okay. Also, this negatively charged particle is having a positive functional group. So let me tell you once again. This column containing stationary phase is embedded with negatively charged ions, and these negatively charged ions are attached with positively charged functional groups. So when we place or pour the sample mixed with buffer in this column, if this is cation exchanger or cation exchange chromatography, these positively charged functional groups will be replaced by new or powerful or strong positive charge of the sample, and these functional groups will be eluted out. So if this is a cation exchange chromatography, the cations, strong cations which are present in the sample will replace these functional cations and will be replaced with the new cations present in the samples. The functional group cations will be eluted out from the column, which can be detected in the detector. Now let me talk about anion exchange. If this is the column and this column is filled with stationary phase, that is resin, and it is an anion exchange chromatography. So these resins will be embedded with positively charged ions, and these positively charged ions are having counter functional groups, negatively charged functional groups. So if the sample with the buffer is poured in this column, the strong negatively charged functional group will replace this negatively charged strong negatively charged ions in the sample will replace this negatively charged functional groups. So this is anion exchanger. So the parts of the ion exchange chromatography are, it is having reservoir, column and detector. So this is the column containing the resin. This is the reservoir, as I mentioned earlier, containing the sample with the buffer. And this is poured inside this column, and the unwanted things are eluted, which can be detected in the detector, and a pictorial graphical representation is formed or can be seen in the computer. So these are the parts of the ion exchange chromatography. Now let me discuss in brief. This reservoir is attached to a tube-like structure called the column. This is the column. Reservoir is filled with mobile phase, which is the buffer solution. Sample is mixed with the buffer solution and it is run through the column. Now the column consists of resin. These resins are having fixed pore size. Inside the column, stationary phase is there. Stationary phase here is the solid adsorbent material. It is made up of resin. Resin consists of beads, which consists of charged particles. If the beads contain negatively charged, it is called cation exchanger. So we should remember, if these beads contain negatively charged, it is cation exchanger, and if these beads contain positively charged, it is called as anion exchanger. because in cation exchanger these bits contain negatively charged but it is having positive functional groups, and in anion exchanger these bits are positively charged but it is having negatively charged functional groups. So the eluted things are detected in the detector, and this column is attached to a detector and the detector is attached to the computer. The basic steps in ion exchange chromatography is sample is mixed with buffer and it is run in the column. The desired things are obtained as elutant or eluted out, which can be detected by the detector. So, 4 steps: equilibration, that is buffer to check packing, that is if this is the column, equilibration means to check the packing of the resins. Loading, sample to be purified plus the buffer at specific pH. Washing, to wash the unwind or unwanted components using buffer. Elution, to elute the bound sample. So these are the 4 steps. Ion exchange chromatography is basically of 2 types, anion exchanger and cation exchanger. So like any other chromatography, there are 2 phases. One is stationary phase, another is mobile phase. And this is same with both anion exchanger and cation exchanger. So let me first talk about anion exchanger. This is the column and it is attached with the detector, which is further attached with the computer. So in anion exchange, the stationary phase resin is embedded with positively charged ions, which is further attached with negatively charged functional group. So when the sample with the buffer is poured in this column, the sample with higher strength of negativity will replace these negative functional groups and will bind with these positive ions. Similarly, in cation exchange, the sample with the buffer with higher strength will replace these positive functional groups and will attach with these negative ions in the resins. So in ion exchange chromatography there are 4 stages: equilibration, loading, washing and elution. In equilibration, what happens, the equilibration buffer is poured in these columns to check the packing. So if the detector gives a straight line, it indicates that the packing has been done correctly. Similarly, in loading buffer is used with the sample, mixed with the sample to be poured in this column. And one thing, in equilibration, the flow rate is fixed, but in loading the flow rate is relatively low because to make sure that the sample is properly bind with the ions in the resins and will replace these functional groups. In washing buffer is used to wash the unbound molecules, and the detector will show some readings. And the last elution, it is done with either salt or pH buffer with different pH depending upon the type of nucleic acid or protein. So elution buffer is used to get the purified protein or the enzyme. So this is how ion exchange chromatography is done.